Everyone, welcome back. So for today, we should be talking about protein degradation using proteasomes. As we all know, proteins are among the most important substances in the body. They are large complex molecules made up of amino acids and are used in different bodily functions. But even though that they are very important, they also need to be degraded, especially when there is low energy, when there is abnormality with our enzymes, or even to regulate our bodily function. During periods of low energy, proteins have to be broken down because they can be glucogenic or ketogenic. They can produce glucose or ketone bodies depending on their structure. When enzymes, which are made up of proteins, are having abnormalities, they have to be broken down to prevent further damage to our cell. There are also some enzymes that can only be regulated if they are broken down. So protein degradation takes place to regulate some of our enzymes and their activities. So, proteasomes are among the ways that we can degrade proteins. It is a selective and ATP dependent pathway, and it is also known as the body's recycling bin of proteins. It is called ATP dependent because you need ATP for it to function. It has to be coupled with ATP. It is also called selective because only enzymes that are labeled with ubiquitin, which is the red circles in this diagram, only enzymes that are labeled with ubiquitin are going to be able to be degraded by proteasomes. So as a quick recap, let us see how ubiquination takes place from the previous lesson. So this is our ubiquitin. It is activated by this enzyme. It is activated by getting rid of this oxygen bond here and replacing it with the sulfhydryl bond of the enzyme to form this activated ubiquitin. This activated ubiquitin is going to have an enzyme transfer. It's going to be transferred from enzyme 1 to enzyme 2 to create this another activated enzyme. From there, this second activated enzyme is going to be bound to the protein, which is this brown looking object in this diagram, by enzyme 3, the third enzyme. So it would form an isopeptide bond with the protein, with the lysine group of the protein. So that is how the protein that we want to degrade is ubiquinated. So let us go back to our proteasome. Proteasome is a multi-unit complex that is composed of a core 20S and 19S subunits. The 19S subunits could be found at either ends of the 20S subunit. It can be found on 1, on 2, or even in both sides as shown in this diagram. So the 19S subunit is called the regulatory subunit because it allows for it is responsible for filtering which proteins would enter the actual catalytic core, which is responsible for degradation. The 19S subunit looks for the condemned protein by looking for the ubiquitin. So let's just draw a quick ubiquitin protein here. So this is how it looks like. Ubiquitin is the one in red. So this ubiquitin molecule is bound to the ubiquitin receptor in the lid of our regulatory subunit. So the lid contains an ubiquitin receptor that binds to the ubiquitin and holds it in place. While it is bound in place, our base contains ATPase. So it contains AAA ATPase that unfolds this protein. If you guys could notice, this protein is very, very big. It is multimeric. It has a different conformation, which could not necessarily fit within the small cavity of our enzyme. So we need an ATPase that would unfold it to make sure that it turns into a noodle and enters our cavity. The cavity, which is the 20S catalytic core, which is the one that is responsible for degrading the enzyme, it contains an alpha and beta subunit. So the alpha and beta subunits form a cavity inside when it is stacked together. So the 20S subunit is called the catalytic core. It contains a isopeptidase that removes the ubiquitin molecule from the protein. So it removes the ubiquitin molecule to ensure that it is not degraded. Ubiquitin is also a protein, so if it is not removed, it would also be broken down, which we don't want. We want to make sure that ubiquitin is not broken down so that it can be reused for our future labeling of other molecules. Inside the 20S subunit, we also have nucleophiles. We have nucleophiles that attack the isopeptide bond of the proteins and break them down. So this molecule right here, would enter the ubiquitin and it would exit out as small peptides that are about 7 to 9 amino acids in length. So the ubiquitin molecules are also released. So as a short recap of what happened, so we have a compound, a protein here that is condemned. It is condemned because it is labeled by our ubiquitin molecules. So our ubiquitin molecules, our ubiquitin labeled protein will be recognized by our lid, particularly the ubiquitin receptor in the lid, and it is unfolded by our ATPase within the base. And after that, it is unfolded and enters the catalytic core where it is broken down by nucleophiles. And after that, it would release our peptides that are about 7 to 9 amino acids in length, along with our ubiquitin molecules. Within the cell, the amino acids, the peptides are broken down to amino acids which can be used by the body. If the cell needs energy, the amino acids can be used to make ketone bodies or glucose. But if not, they can be used to create other enzymes or other purposes the cell needs it for. So that is all. Thank you very much, and we hope you have a great day.